Welcome. Today we're going to be talking about oncogenes, and I have this lecture split into two parts. In the first half, we'll do just a little bit of introduction into exactly what oncogenes are, and then we will talk about some of the mechanisms that result in the production of oncogenes. In the second half, we're going to dive a little bit deeper into one specific oncogenic pathway that's commonly found in a lot of cancer cells. All right, so I think we've talked about this at least briefly in some previous lectures, but oncogenes are essentially mutated, hyperactive versions of normal genes called proto-oncogenes. So proto-oncogenes would be any gene that basically helps positively regulate cell division, right? Helps drive cell division. So if a proto-oncogene acquires what we would consider a gain-of-function mutation, meaning it either takes on excess function or possibly even gains a new function, that could result in that proto-oncogene becoming an oncogene, which could then in turn help drive a cell into the cancerous state. And just to remind you, right, we could really have two different types, or I guess outcomes of mutations. So mutations could cause a gain of function, as we just talked about, or a mutation could knock out or essentially break a gene. And in the case of a proto-oncogene, a loss of function or knockout mutation is not going to be a problem in terms of cancer. But when we talk about tumor suppressor genes in the next chapter, we'll see this scenario is reversed. Okay, so how were oncogenes first identified? And this came from what now seems like a very simple experiment, but was actually a really big hurdle at the time. Experiment conducted by a famous cancer researcher named Robert Weinberg. Um, and he had the really brilliant idea to take DNA from human cancer cell, chop it up into very small pieces, and then inject just one or a couple of those small pieces into normal cells being grown in culture in the lab, in this case mouse fibroblast cells, um, and look to see if any of those injected DNA fragments would cause those cells to become transformed into cancer cells. And that would be verified by injecting them into mice to see if they could form tumors or not. And if we did see a transformation process, what Weinberg did was then go back with his research group and try to identify, okay, well, what genes are present on those DNA fragments that cause this transformation. And by doing this process, they were able to identify the gene RAS, which was the very first oncogene found. And of course, now we have a long list of oncogenes uh, that have been well studied. But that was RAS was the first, and in the second half of this chapter, we're going to dive into the RAS pathway and focus on that specifically. All right, so as we've already discussed, you can take a well-behaved proto-oncogene, give it a gain-of-function mutation, and convert it into a oncogene. And there's 5 mechanisms by which this process could happen, and I wanted to go through these one at a time. They're listed here. And we'll start with point mutation, which was the first on the list. And I know you're aware of point mutations is referring to the fact that you were just mutating a single point in the DNA, maybe just a single nucleotide substitution. And in this particular example, that single nucleotide change just in one codon in the RAS gene is causing a missense mutation, where we're ending up with a different amino acid in this particular point in the RAS protein itself. And in this case, this one changed amino acid is enough to give that RAS protein excess activity. Okay, so just a simple point mutation, if it's in the wrong place in the gene, could have this particular effect of turning the proto-oncogene into an oncogene. The second mechanism for creating oncogenes is what's called gene amplification. And as that name suggests, we are literally just amplifying the number of copies of the proto-oncogene that are present. So we're not actually mutating the gene in any way, but in creating excess copies, we're then producing excess oncoprotein, right? So we've got that hyperactivity essentially, so we're still crossing over into oncogene territory. And there's a few different mechanisms by which gene amplification can happen. Uh, the mechanisms are actually pretty complex and not entirely understood, but we do know that these three different phenomena occur with some regularity. And so I wanted you to be aware of these. The one on the left here is what's called double minutes. And this is essentially extra chromosomal copies of the proto-oncogene. So the images you're seeing across the bottom here are basically a fish fluorescent in situ hybridization, where they're using this green fluorescent tag to label copies of a particular proto-oncogene. And excuse me, what you see here in the double minutes example is all these little green dots outside of the blue chromosomes. These are extra copies of that particular proto-oncogene. And you can see there's a lot of them, right? We've got a lot of amplification going on there. So if those copies are being transcribed and translated, we're getting too much protein. The middle example here is what's called HSR amplification, which stands for homologously staining regions. And this is basically where you're getting multiple copies of a proto-oncogene produced right next to each other in a single chromosome. So this sort of makes a bit more logical sense, right? If you're going to overproduce copies of a gene, that they would just kind of stay right next door to each other. And then the last example is what's called distributed insertions. In this case, we do have multiple copies of that proto-oncogene, but they're scattered across numerous chromosomes in the genome. And as I said, we don't really totally understand the mechanism, but we do know this is typically due to mistakes during DNA replication, where the DNA polymerase will get stuck replicating a particular gene or segment of DNA over and over again. One common proto-oncogene that's known to be amplified in a fair number of cancers are various versions of transcription factors that are part of the MYC gene family. And just to give you one example here, this MYC-N gene is known to be amplified very commonly in neuroblastomas, which are neural crest tumors usually found in the adrenal glands. And just to show you that gene amplification can really be directly linked to the severity of a particular cancer. So in this case, we're looking at survival percentages in relation to how many copies of the MYC-N gene are known to be in that particular tumor. And so we see if there's no gene amplification, so just the normal 2 copies of the MYC-N gene, we have, let's say, 65-70% survival percentage. Whereas if we have 3 to 10-fold amplification of that gene, we drop down well below 50%. And if we've got more than 10 copies, very low survival percentage. So even clinically, right, just the number of copies of a proto-oncogene can be very significant. And I just wanted to show you this table just to point out that there are actually lots of examples of oncogenes in human cancers that are known to be amplified. So these are all different human oncogenes. We can see a couple of RAS genes. Here's some of those MYC genes, cyclins, CDKs, some things we've mentioned in previous chapters. All amplified in a wide variety of different human cancers. So it's a common phenomenon. The third mechanism I wanted to mention is chromosomal translocation. I know you're familiar with that basic idea. Um, and there's a couple of ways chromosomal translocation could result in oncogene formation. One unique method is through the formation of what's called fusion genes or fusion proteins. And I know some of this came up in some of our case study examples, so hopefully you're at least somewhat familiar with this idea. But this particular example is a Philadelphia chromosome, which is a common chromosomal translocation found in certain leukemias. And this particular chromosomal translocation is between chromosomes 9 and 22 and involves breakages right in the middle of these two genes, the ABL gene on chromosome 9, the BCR gene on chromosome 22. And when these chromosomal pieces swap places, over here on chromosome 22, we end up with this BCR ABL fusion gene that produces a fusion protein that actually has some biological function. Um, so, we're fusing these two genes. That results in a production of a fused transcript, which results in production of a fused protein. And if that sort of Franken protein, if you will, actually has some biological activity, right, that could create a oncogene. And you can imagine this is fairly rare, right? The fact that you could take two genes, just chop them in half, slap them together, and actually get some activity that's driving cell division seems somewhat unlikely. Um, but that being said, it does happen with some regularity, particularly in leukemias and also some lymphomas. Um, and we think the reason for this is because uh, B cells, right, blood cells that are responsible for producing antibodies, um, they go through a process called VDJ recombination to produce the huge number of different antibodies that your body might need to make. And because there's a lot of this recombination going on, apparently this can result in, an excessive number of translocations happening at the same time. And just due to random chance, at least some of those translocations may result in the formation of these fusions between genes that could then help drive cancer, as we just talked about with the BCR-ABL example. Another way chromosomal translocations could result in the formation of oncogenes is just by moving a proto-oncogene, in this case MYC, um, from a chromosomal location where it is expressed at a low level to a new location where it's expressed at a very high level. And so this is the chromosomal translocation that is commonly seen associated with Burkitt's lymphoma, which you may remember we talked about back in the virus and cancer chapter. But we're moving MYC, it gets overtranscribed, we produce too much of the MYC protein, and are into oncogenic territory then. And so, again, we see this happening with some regularity, particularly in leukemias and lymphomas for the reasons I just talked about. But there's quite a few well-known examples of translocations causing overexpression of a proto-oncogene. Fourth mechanism, DNA rearrangements. I'm going to talk about insertions here separately in mechanism number 5. A deletion could cause production of a proto-oncogene if you were deleting a segment of DNA that maybe had some restrictive, repressive effect on transcription, right? So you remove that repression and end up maybe overexpressing a proto-oncogene. Transposition, moving a gene to a new location, inversion, also, moving the position of a gene could cause overexpression for reasons we've already talked about. Or inversion could lead to production of Franken proteins, fusion proteins, just as we talked about with translocations. So in this one particular example, getting this inversion of this segment of DNA here, and it just happens to involve breaks in the middle of these two genes, one called tropomyosin 3, which is important for muscle contraction. This gene and its coded protein has nothing to do with cell division, really, or cancer. But the NTRK1 gene is a growth factor receptor, right, which is involved with cell proliferation. And so if we invert this segment of DNA, we produce really two fusion genes. The one on the left here has this big chunk of that growth factor receptor, a little piece of the tropomyosin gene, and that will encode this protein that has excessive, basically, growth factor receptor activity. So it's essentially constitutively active, always on, produces that oncogene. All right, then the last mechanism I wanted to mention, insertional mutagenesis. And again, we saw this in the chapter where we talked about viruses and cancer. But if a viral DNA is inserted next to a proto-oncogene, because the viral genes are being transcribed at a very high level, that will typically drive higher expression of other nearby genes. And in this case, the nearby proto-oncogene, MYC, gets overexpressed and becomes oncogenic. So just to summarize what we've talked about so far, we've got our well-behaved proto-oncogene, and by really any of these five mechanisms, we could create a scenario where we're either producing just too much normal protein activity, so hyperactive protein, or possibly some new function. So I will stop there and I'll pick this up in part 2.